Hi everyone, welcome. We will start in around 5 minutes, and I shared the paper and the presentation on top of the room and the paper link in the chat, so feel free in the meantime to check it out. And Dr. Florindo, she will be joining us in a few minutes, so we start on top of the hour. Thank you. Oh, and feel free to share the room if you think this is something that interests people. So thank you so much. Hi, Arpad. How are you? Feel free to come up to the stage if you want to be part of the discussion in a few minutes. Thank you.
Hello, I just come keep you company until you start.
Thank you for coming. I think it will be really interesting, LT, and thanks everyone. We'll start in a little bit. App just broke down. I was sharing the room on LinkedIn and the app just stopped working. So thank you, LT, that you came up here. You saved the day.
What happened? You got kicked out of the room?
Yes, I was sharing the room on LinkedIn and I just got kicked out.
Yeah, I
While
think
doing
it happened a couple of times yesterday in a house. The host who started the room kept popping in and popping upstage, downstage, upstage. So maybe that's it. Hopefully nothing will happen when we start the real talk.
Yeah, I agree. Hi, Joyce. Hi, Victoria. We'll start on top of the hour, so thanks for coming.
Yeah, hi. I may be in and out, so but I wanted to support the room. Great. Look forward to this topic. Sounds very great and interesting, and it's really important to make things lower cost.
Yeah, I agree that this is like really interesting because of that. And I think, I really always like when it's really elegant research, but also it's something that people in the future can afford to use. So that's the perfect situation. So we'll Dr. Florindo, she is trying to get in, and she's new to Clubhouse. She's joining Clubhouse for us, so please have a few minutes of patience. And if you in the meantime want to check out the presentation and the paper that I shared in the chat, feel free to do so and share the room while we wait for a few minutes until Dr. Florindo, she can join us here on stage. And thank you, everyone.
Yeah, I came at the healthcare area from the patient perspective. And I'm also come from my mom always said she was a depression child, so she didn't like to spend money if she could avoid it. So I've always been like very frugal. So I love things that pay attention to the cost.
Hi, Lena. How are you? I'm so glad you made it. Thank you. So the unmute button is all the way on the bottom right. If you press that little microphone symbol, you should
Okay,
be
now
able.
yes.
Yes.
Now I think you can hear me. It's okay if I don't see you, right? Or not?
Yes. Exactly. That's just the. We have your picture and your voice. So.
Okay, good.
Yeah. Thank you so much for coming, and I'm really thankful that you went through the trouble to making the account and everything. So thank you for that.
Thank you for the invitation. It's a pleasure.
Yeah, and before we start, I'll give a short introduction, and we usually have like a couple of interview questions, if that's okay with you, and then we go
Sure.
ahead and dive into your very interesting research. So, yeah, welcome everyone to the Science Society, and a special welcome to you, Elena. And yeah, as I said, I wanted to introduce you to the audience here. Um, Dr. Elena Florindo, she is a group leader at the IMED Research Institute for Medicines at the University of Lisboa. And she did her she went to school in pharmaceutical sciences at the Faculty of Pharmacy at the University of Lisbon in Portugal, and then she did her PhD in pharmaceutical technology, nanomedicines, and vaccination at the University of Lisbon in Portugal, in collaboration with the University College of London in the UK. And she did her habilitation in pharmaceutical technology at the Faculty of Pharmacy at the University of Lisbon. And she is now the Associate Professor and Faculty of Pharmacy at the University of Lisbon. And her research in her lab focuses mostly on the characterization of mechanisms of cellular dynamics and their crosstalk and networks to identify new targets, to engineer translational nanotechnology based systems for drug delivery, imaging, and immunotherapy for clinically relevant problems like cancer, inflammation, infectious, and genetic diseases. And yeah, as I said, before we start, we usually have like a couple of general interview questions so people get to know you a little bit better. So, usually, our first question is, how did you choose the path of science? Was that something you always wanted to do, or was there maybe a book or a teacher or professor that kind of sparked your interest in the field of science? Thank you.
Thank you, Katerina, for the kind introduction. Regarding your question, I think I always thought about myself in something related to science, but it is true that around like a teenager, like 15 years old, I always liked to write a lot and learn. So at some point, I was debating between which path to take, if it was mostly related to science and health, or to go in a completely different one, towards like a law. But then for me, it was always much more when we had to make that decision. We are very young, but still, so I thought that the path that was more interesting to me, and something that I'm very curious, so and I like surprises a lot. It was more into the science. So I started to think about which would be the best way. So debating between the medicine and pharmaceutical sciences, because I also would like to make then the medicines to treat the diseases. So was more or less like a continuous path.
Yeah, that's very interesting. That yeah, you followed curiosity and also passion. That's really wonderful to hear. And you kind of answered already how you chose your field of research. So, is there maybe for this project or for projects you started doing then in your own lab, is there maybe like a background story? Was it really easy to get funding? Was it really hard? That we had researchers here sharing.
Yeah,
Everyone
exactly.
told me I was crazy in the
Yeah.
beginning. So, is there maybe something there?
When I was doing my PhD in London in the School of Pharmacy, my PhD was to develop a vaccine at the time for infectious disease, but it was a veterinary vaccine, so it was for a horse disease. And at the time, it was, I was already thinking and understanding from my supervisors at the time. It was Professor Oya Halpar in the University of London and Antonio Almeida here in the University of Lisbon. You have from our national agency the opportunity to have like joint PhD programs. So in here in Portugal, it was definitely much more difficult than it was in London, but especially because Oya was already um, really advanced in her career, and she was getting a lot of funding from the Ministry of Defense and the WHO. So but at some point I understood that infectious diseases was not the easiest way to attract funding, but I also liked cancer. And as I was working in immunology, and at that time it was the when you start hearing about the Ludwig Cancer Institute in New York and also in Brussels, trying to understand which were the markers in the tumor that were related to immune response, which could be strong or changed in a way that would awake our immune system towards that cancer cell. So I thought, why not to merge what I was doing, but instead of looking into infectious disease, to go for cancer. And it was clear that cancer would attract much more funding, even though, yeah, you are right, funding is not easy. We have a lot of rejections. I remember I always wanted to work on the vaccination for cancer, and there is still, so even today, so many people that are skeptical about it. And at the time, people in my institute, they were always saying, no, 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 go to work on that, but focus on gene delivery, for example. sRNA delivery, it's much more fancy, trending, it will be much more easy, it will be easier for you to get funding. Then I think the key was that I started from the very beginning to work on projects that were collaborative. I always understood that we needed to have people that would help us with their knowledge in fields that we are not comfortable with it, or we it's not our expertise. So I started to apply from the easiest way out, which was to what we knew more at the time, which were our projects from our national agency, already in collaboration with, for example, Véronique Préat from the Université Catholique Louvain in Brussels. And I think I was lucky because I was awarded that project 2 years after I finished my PhD. And I was also awarded PhD fellowships for my students. So it started from there. But then we get the real life. And it's true, we have a lot of rejections, but I try to think about these rejections in a way to improve, or that we didn't exactly describe what we think that we were describing, or the way that we were describing was not clear enough, and try to make it better and never give up, going there again, making the right connections, or at least people with whom you feel good working with, that are complementary with you. So I think this is the path that I've been taking. And I think even in Portugal, with all the problems that we have in terms of accessibility to funding, it has been a nice journey, I think so.
Well, I'm so glad you keep taking the path of, seeing it kind of in a way to improving and from this positive way. That's wonderful, and congratulations for that. And
Thank you.
also for this really amazing research that you publish and have been doing. So that's really wonderful. And yeah, we have pinned your presentation on top of the room so people can access it. They scroll through it. So people here on the app, they have to kind of scroll through it by themselves. So it's really helpful if you refer to the slide number when you switch slides. And
Okay.
yeah, the stage is yours. And thank you again, Elena.
Okay, thank you. Just to tell the audience that please feel free to interrupt me and make questions whenever you want. I talk a lot, so this is a problem sometimes. So in the slide 2 that you have there in the presentation, this is not new for none of us, I guess, which is we are all aware about the revolution that the immunotherapy brought into the cancer treatment. But still, and even though it is clear that it brought some solutions to patients that didn't have any other option, like metastatic melanoma patients. But when we look at the numbers in a less positive way, we see that still a huge percentage of them do not respond to the therapy. We are talking about around 70% of melanoma, also in breast cancer. But still, the 30 and 20% in breast cancer, for example, that are responding, it's really important. But this shows that we have very good drugs, and these monoclonal antibodies against immune checkpoints are very powerful drugs. But we always thought that maybe there is still room to improve them, or at least to find a way. This was how this project started, to create these targets in tumors that were not there, where these targets are not present. So we were trying, for example, to use vaccination for that. And then we thought that, okay, but there are still these tumors like melanoma that express nice levels of PD-1, but still PD-L1, but still they do not respond. So is this just due to the other components of the tumor microenvironment, or we need to target other receptors to complement this response, or we can still take advantage of this anti-PD-1 and try to improve the efficacy of these antibodies. So in the slide 3 is just a summary of what I was saying, that these PD-1, they are expressed by T cells. The PD-L1 and the PD-L2 are the ligands of this receptor. They are present on the tumor cells, also in other cells, for example, dendritic cells, endothelial cells. But the problem is that when this PD-1 binds to the PD-L1, the T cells, they do not expand into effector, immune effector cells, so they are not active. So if you go to slide 4, it was already proven that if you use an antibody to block this connection, then these T cells, they become activated, and they induce the destruction of this tumor. But still, you have several problems here. The response rate, it is what we have just saw, but also there are another two problems that are really important. That is the resistance. So this means that the patients at some point, they stop responding to this therapy. but also, and is equally important, are the adverse effects. And this was something that caught our attention because even if you have a really nice therapy, really effective as and strong as these antibodies, but you as a patient cannot tolerate this therapy, then we have a problem because we have a very good solution, but the patients cannot use them. So what we thought it was, okay, so let's find a way if at least we can take advantage. And then if you go to slide 5, you have like a summary of what we would like to have, which is a molecule, a small molecule. So we are talking about something below 500 Daltons. If you compare with the 2000 of the monoclonal antibodies, for example, something that it could anyhow, besides being small, to accommodate in the receptor of the PDL1 and inhibit its interaction with the PD1, but at the same time something that you could then formulate. And this, as a pharmacist, it was something that we know that we can always take a drug that may be toxic or may not have very nice pharmacokinetic properties, but then you formulate it as a pill or a capsule, and you can take advantage of it and overcome some of the adverse effects, or for example, not getting it into contact in the stomach and getting into the intestine and being absorbed. So when we started to work, and even now, there were no small molecule modulators to change the activity of these immune checkpoints or others. None was approved. Now there are 2 in clinical trials, which is a very good news, because this is also something that you need to fight. It's not only to show your science, but also to convince that this is something that is not science fiction, is real, and you can really take advantage of it and bring it to patients. So in slide 6, you have a picture of Rita. Rita Curcio was a PhD student in my lab that, together with a colleague of mine, which is Rita Get, she's an expert in signal chemistry and computational, and with our collaboration as well with Professor Ronit Sachifainaro in the Tel Aviv University in Israel, we were working in these cancer projects together, and we thought, why not to try to understand, to figure out if it is possible at all to identify a small molecule, um, but in a faster way. And this is something that we always know. And if you go to slide 7, we know that this path about drug discovery is too long. It takes too long, it takes many years. And at the end, you need to move back and forward in order to optimize your molecule, but everything then synthesize it at a large scale, go back because it didn't have drug-like properties. So this would take ages. And Rita, what she thought, it was, okay, so let's do it computationally. So we took the knowledge of the 3 of us to understand first if we could identify using this computer-aided drug design. So this means that you take advantage of the knowledge that we already have about the crystallographic structure of this protein or others that you want. And by predicting the molecules that would bind to those receptors, we move fast and we can identify potential candidates that will go directly to the ex vivo and in vitro studies. And at least we would discard those that at the end they are not active at all. So if you go to slide 8, this is a summary of what we did. So Rita started, and Rita Curcio was a brilliant PhD student. She came from chemistry. Then she started working in immunology in my lab. And then she learned computational. So she did all these tasks. She started looking into the structure of the PDL1 protein. At the time when she started, it was available only 1. Now you have at least 3 available, which is good, because then the more details you have in the crystallographic structure of these proteins, better you can predict the binding of the molecules to the pockets that you identify as being essential for this interaction between the PDL1 and the PD1. So after knowing this, as I said, we need to take those small molecules that we identified and do a kind of a screening in vitro just in a plate. That this plate is coated with a PDL1. Then you incubate with your compounds, your candidates, and you add another antibody that it will be PD1. And then you will see if this interaction is blocked, it means that your small molecule was able to prevent the binding of the PD1 with the PDL1. This was done in a plate. And then we move on to the cells, patient tissues, and at the end we validated in an in vivo model, and I'll show you a summary of this. So if we go directly to slide 10, this was the approach we took for the computational design, computer-aided drug design. So we took a parallel approach where first we knew already that there were some small molecules that have been published in the past um, as being able to interact with the PDL1, but these small molecules were reported as lacking drug-like properties. So, and this prevented that you could use them in the future, even for in vitro studies. We didn't find at the time any studies about the biological effect of these small molecules. But still, if there was knowledge about them and how they interact with the pockets, this was great. So we could use them to understand which were the spots that we needed to block in order to inhibit this interaction between the PD1 and PDL1. So this was the ligand-based CAD. But at the same time, we wanted to also take into account what I said before, which were the crystallographic structures of the PDL1. And for that, we used the structure-based CAD, which was looking at the structure, identify the spots, and using molecular dynamics to understand if these small molecules, even being small, they are able to stay there between the pocket. So, but to do this, you also need to define some targets, right? We cannot just simply go and select all of them that will bind there. So in slide 11, these are the factors that we defined. So the first one, we wanted a small molecule. So as I said before, around the top, it would be like 500 Daltons. We have here 300 to 600. Then this gold score, it's a measure that indicates you if this binding occurs to the PDL1 small molecule. And after you select it, here it was around, if I'm not mistaken, around 800 molecules. And then Rita took those that were the top hits and she observed visually how they would bind to the PD1. So it was a kind of, even if it is a computational, it was also, it had also this component of visual inspection that was really important to then select those that were staying there within the pocket. Because if they are not there with high affinity, then if they bind, they will be washed. So, and the fourth factor, it was we wanted a drug that would also have properties. I mean, they were with a partition coefficient that would be adequate for in the future to have an oral administration of these small molecules, as this would be brilliant, as we would avoid the injection of the antibodies. And also we can play, as I said before, with the formulation to improve the tolerability of these small molecules and also to direct them. And if needed, one of the options that we had and we were considering, but at the moment we don't think we will need it, it was to use, for example, then drug delivery to just target these small molecules to the targeted tissue, like the tumor site. But we want to avoid that and to have the simplest system as possible, to be also affordable. So after all of this, and you can see in slide 12, if you click on it, you have a movie, and this movie shows you exactly the purple. You have a molecule that is yellow, then it comes, if I'm not mistaken, orange, a purple first and an orange after. This purple is the 69 molecule that we then used for the in vivo studies. And this was the kind of visual inspection that Rita did, and it was important to select them. Then in slide 13, you have just a summary of all the compounds, and here we are talking about roughly 100 compounds that Rita selected for the in vitro and assays, for the screening in vitro assay. And this was important, for example, the molecular weight, the partition coefficient. So you can see that we had molecules that were very small, others that were above the threshold, like the 500. So these ones we didn't pursue with them. But it was nice to see that we actually had different molecules that were predicted to bind with a high affinity to the receptor, and with some of them more hydrophobic, more hydrophilic. And in this case, you want some lipophilicity, because ideally we want these small molecules to cross the biological membranes. This is important, and it would be an advantage compared to the monoclonal antibodies. They cannot do, and this would give advantage of this small molecule, for example, if you want to target brain tumors. As the antibodies, they don't cross the BBB, and these small molecules, if they have some lipophilicity, they they can diffuse also because they are small. So from these, we took these 100 compounds, and we did this heat validation study, which was the screening I was describing before. So you just have like ELISA plates where you have the PDL1 conjugated to the well, and then you add your small molecule, and on top of it you add the antibody that would bind to the PDL1, so the an anti-PD1. If you manage to block this interaction, then you get a lower level, a lower percentage of this interaction. And you can see in this slide 14, in green, in the upper graph, you have those that were the best ones. Here we selected the threshold around 25, but you could select more. But as we had so many positive hits, it was already for us really nice to go for those that had the highest ability to prevent this interaction or to block this interaction. So we selected 12 compounds. We performed, and this you can see in slide 15, the dose response curves. This is also important because, especially if you look into the translation of these compounds into the clinical after, which you need a compound that is able to block this interaction, but you need also a compound that is able to block this interaction at a lower concentration. So you have already, and this is a challenge, to have compounds where you can have this efficacy at the nanomolar. This you can get after, and if you remember from our paper, ours is like in the hundreds nanomolar, like 0.1 micromolar, which is not bad, but is not brilliant, I understand. But then we pursued with this one because it was highly stable, and this molecule has a really room for improvement in terms of changing of the chemical entities in the molecule. And this was what we thought when we pursued with the 69. We had others that were also promising, like the 47 and the 42. If you see in these graphs, they have a dose response curve very similar to this BMS 202. This BMS is one of those small molecules that has been developed by Bristol Myers Squibb, but at some point they just stop using it, saying that they lack drug-like properties. So most probably they were not stable or lipophilic enough, or they were too lipophilic, so you couldn't work with them. So in addition to the 69, we had the 47 and 42. They were with different scaffolds. We had to select the one of them to pursue, and so our first candidate was the 69, as it was the one that had a dose response curve, um, also similar to the BMS 202, but it was highly stable, the molecule, and with room for improvement chemically, I mean. So in slide 16, and then was the other question, which was we showed in the plates that it binds, but it's not enough. We need to show that it really binds. So at this point, we thought about two assays to do this. So the first one is what you have in slide 16, which is you have your protein, the PDL1, you mix your small molecule with the PDL1, and you increase the temperature. So this melting temperature, and if you measure the fold change on this temperature shift when you have the small molecule complexed with a protein, it means that your complex is stable, which means that it binds with a higher affinity. So this is what this assay showed us. Again, the 42, the 47, and the 69 as well. And then in the next one, slide 17, it's a more accurate way to show this. You could also do it by crystallography, for example, but we used NMR, where again we mix the protein with a small molecule. In this case, we use the SEM69 you see in C and in D. And also we use the BMS 202 as a positive control to show if the change that you would have on the NMR spectra would be similar. And you can see that B and D, which is the spectra that you have when you have the combination of the BMS 202 with the protein anti-PDL1, is similar to the one that we got with a small molecule. So this was the proof that the small molecule binds to the PDL1. We don't know if it is the only mechanism by which we got the activity, but for sure, at least, is mediated by the PDL1 interaction. So next, we wanted to understand, okay, you want to inhibit this interaction, but we don't want a small molecule that will be cytotoxic. We don't want it to use as a drug that will kill the tumor cells and everything else that it finds on the way. So we did these very simple studies where you just study the impact of these small molecules in the viability of cells. We used 3 cell lines, one breast cancer, one melanoma cancer of humans cell lines, because all our study in terms of the computational, it was done using the PDL1 human structure, which is slightly different from the mouse. The mouse would be very interesting for the preclinical studies, but we really wanted to move forward for the human setting. So you could see that some of these molecules, they did in fact had a negative effect on the viability of the cells, but again the 69, the 47, the 42, and some others, they didn't. So these were good news. So they were binding and they were not killing these cells, either if they were cancer cells, but also we had as a control endothelial cells as well, which you have in this slide 18 in the bottom. So we move forward and we wanted now to understand if these cell lines would confirm the data that we got in the plates, which would be the inhibition of this PDL1. So for that, we just cultured this cell line, the breast cancer. We added our compounds and we measured by flow cytometry the accessibility of this PDL1. And again, it confirmed that they were in fact inhibiting. But here we started already to see a difference compared to the BMS 202. But you can see in red in this slide 19 the the effect of the monoclonal antibody, and you can see that this really has much higher affinity than the small molecules. But it's okay, we were expecting this. This was nothing different from what we were expecting. So if you go to slide 20, here we can see then the idea was to understand, okay, we saw in vitro in a plate that inhibits, then we saw in the cells in vitro in a cell line of breast cancer that it also inhibits. But we now wanted to see how it would be the interaction between these tumor cells when you inhibit the PDL1 with your small molecule. What would be the impact on the T cells? So in this first approach, we followed a stepwise approach that we increased complexity. So because we wanted to discard those candidates that would be like false positives. So what we did, we used the same cell lines. We used human PBMCs, where you have mostly T cells. We co-cultured them after the tumor cells being activated with the interferon gamma. The interferon gamma is a major activator of this PD-L1, which mimics what you have in the tumor niche when in the presence of cancer. And when we added the small molecules, you can see on the right side, the T cells were able to activate interferon gamma. To secrete interferon gamma, to secrete TNF alpha. But we also saw, and we confirmed that inhibit the PD-L1 on these tumor cells. And in here, some of the small molecules change the PD-1 expression on the T cells, but our 69 didn't change in this assay, but it was okay. This was not our major focus. We wanted to see the effect on the PDL1, and this is what we got. And the 69 was one of those that were changing more the secretion of these cytokines. So now that we confirmed that they have the same effect in the human cells, we followed these collaborations that we have in the hospitals just across the street. In our faculty, we have one of the major hospitals in Portugal, and we have collaborations with some oncologists that they are also researchers, and we got some cells from them. Here the challenge is to have matched tumor cells and blood from the same patient. This is important because we need to have the HLA matching, right? From the T cells that we isolate from the PBMCs and the tumor cells. We got from melanoma, bone metastasis of breast cancer, and metastasis of lung cancer as well. And we did a co-culture of these two populations. We treat them with with our small molecules, and also using the atezolizumab as a control, because this is one of the anti-PD-L1 antibodies used in the clinic. This you can see in slide 21. And this was already a really interesting result for us as well, because if you pay attention to the different slides, in red you have the effect when the cells were treated with the monoclonal antibody, and in yellow you have when the cells were treated with our small molecule. The CD28 was the positive control, and you can see that in all of them, the small molecule, the impact that it had on the T cells, it was similar or even better than the monoclonal antibody. And interestingly, what you can see in B, in the graph PD1, it was that the small molecule was decreasing the PD1 expression. This was interesting. We didn't see in the cell line, we only saw in the patient tumor samples. And this means that somehow the small molecule is inhibiting the PDL1, but it's also having an effect on the PD1, which is important so in a way you are blocking the two targets. Also important, it was the expression of the CD107, so the T cells were activated, and in some cases as good or even better than in the cells treated with a monoclonal antibody. So this was a very good indication for us. For us, it was already enough, but you see that they are activated. But one of the major challenges that these monoclonal antibodies have, as I said before, is their capacity to infiltrate in the solid tumor mass, as I explained, for example, for the membranes. So what we tried to do, it was to actually to see the effect of this small molecule in the ability of these T cells to infiltrate the tumor mass. So this is what you have in slide 22, where we prepared spheroids, again using tumor cells derived from, or cancer cells derived from tissues from patients, and matched PBMCs. We prepared the spheroid with the cancer cells. You treat the spheroid with a small molecule or the monoclonal antibody, and then you add the PBMCs. And you will see if the small molecule or the antibody has any effect on the ability of these T cells to infiltrate in this spheroid, which is a 3D co-culture, right? And what you can see in slide 22, it was really interesting for us, which was you have the first one is the PBS, and in green you have the staining of the T cells. And here we stained only the CD8 T cells, which was our major focus. So you can see that in the PBS some T cells are there. When you treated the spheroids with the anti-PD-L1, uh, there were still some there, but this amount was really increased when you use the small molecule. So this was again confirming what we thought, which was that the small molecule has a higher ability to improve the infiltration of these T cells, maybe because the small molecule itself, it is able to diffuse in the tumor mass. But this was a maybe, and we didn't have any proof. Even if we, in confocal microscopy, using confocal microscopy, we did several sections, and we understood that the T cells were in the middle of the within the tumor mass. So we had to go to an in vivo setting. Here we had a challenge because we couldn't use a simple murine model, because we are targeting human PD-L1, and we are also changing the interaction of the human PD-1. So we used an animal model, which is a colorectal cancer mouse model, MC38. We used the cell line, and this cell line was changed to express the human PD-L1, and the mice that we used, they were genetically modified to have T cells that express the human PD-1. So this was a really nice model, and actually this was the model used when the immune checkpoint monoclonal antibodies, anti-PD-1, were developed. So this was a good validation approach for us. So we did this study where you had the PBS, a group that was treated with atezolizumab, the human monoclonal antibody, and our small molecule. That is true that we had to give more doses, but we higher number of doses, because the half-life of this small molecule is shorter than the antibody, for sure, because the antibody may have a half-life of a month. So, but what, even though it was really nice to see that the efficacy in terms of controlling the tumor growth was similar, this, I don't remember if I said the slide, so it's the 23, the last one with data. And so it was really the same. And on top of it, the variability, it was a kind of better with the small molecule, which means that the tumors presented by some animals were very similar within the same group, and some of them they didn't have tumor after 30 days. And when we took the tumors of those that still had tumor, even if they were tiny, we could understand that the CD8 T cells, this is what you have in G, were at higher extent, at higher frequency than in the tumors of those animals treated with monoclonal antibodies. And again, as we saw in the co-cultures with the tumor tissues, we had a decrease on the PD-L1, but we also had a decrease on the PD-1, which we didn't see with the monoclonal antibody. So this was a very good indication of a small molecule that now it has a room for improvement in terms of the delivery. This small molecule was administered as the monoclonal antibody, IP, and now we are exploiting it for oral delivery with a formulation as a pill, which can, and by coating the pill, we can change the pharmacokinetics of the small molecule and increase the half-life of this small molecule. So this is where we are heading now, and we hope we can have some data and to prove what we expect, which is that we'll have an oral therapy that can be more easily available to everyone, and with much lower side effects. And this is what I had to show you today. And the other slide is just the 24 is just a conclusion, and the 25 is just the people involved in this study. As I said from the beginning, as I always did since I start my group, I try to join efforts with people that will help us to move faster and better in a better rational way. And in this study especially, you have the role in this, you can see it slide 25, Rita Getz, which is our computational scientist, and Ronitsachi Fainaro, which was in the lab where they developed these 3D spheroids that then we validated in our lab too. And thank you all for being here and for your attention. And please go ahead and ask some questions in case you have.
Well, thank you so much for sharing this really amazing research with us and for presenting it and explaining um, everything in a way that, even I could understand. So thank you for that. And yeah, if people have questions, please go ahead and ask and flash your microphones. Yeah, Dr. Shah, please go ahead.
Yeah, thank you so much, Doctor, for sharing your amazing research with us. So we are aware of the, limitation, and as you just mentioned, one of the reason can be the heterogeneity of the tumor. And my question from you, because we need a preclinical as well as the clinical result, and what exact pathway you were focused on, because we know about the changes specifically about the post-translational changes on the T cells. And I was just wondering, is there any specific pathway that you just challenged during your experiment or not?
Thank you for your question. No, during we just wanted to to regulate, to inhibit the PDL1 PD1 interaction. So we didn't check further if this had any impact on for example, the transcriptional profile of the T cells. But we are trying to understand because they are more active, and the exhaustion profile is much lower. So definitely we need to understand what is the impact, because this can also help us to find a way to identify potential candidates that we can combine as well in the same formulation.
Also about the, breast cancer, you mentioned that was a ER positive. I didn't
Yes,
notice about the type. Okay.
yeah.
And do you think that you can use it for the gastrointestinal, for example, EBV,
Yeah.
GC?
W- Yeah, we are trying to actually we were combining this small molecule with a vaccine that we developed for CRC and pancreatic cancer. And we tested as we had the models, the small molecule, but in this case we only tested in 3D spheroids. But the result was quite interesting, and what we saw is that we can the same activate the T cells, but they are in a much lower number in the pancreatic cancer, for example. So in that case, when we use the vaccine worked as a Trojan horse, so increased the amount of T cells that we managed to infiltrate in the tumor mass, and then the small molecule did the job after we increased the amount of these T cells.
Thank you.
Thank you.
That's a question.
Yes, go
Um,
ahead.
okay. The screen you started with is like 600 to 800 small
Yes.
molecules, right? So,
Mhm.
and after you fixed on this 69, have you, tried to look for other closely resemble the 69 or even maybe multiplication on 69, just to play
Yeah.
around with it a little bit? Yes, thank you.
Yeah, you are absolutely right, because this is what we are doing now. Now what we are doing is like the novo design, so looking at the scaffold and trying to see if any changes are predicted. This is computationally are predicted to increase the efficacy. At the moment, the 69 is still our major candidate, but we are looking into that, because the main idea it will be to keep a kind of lipophilicity that it has, but is doable to formulate, but still to maybe I think we can even increase the efficacy or the impact that it has in the activity of the T cells by changing some of the functional groups in the molecule. You are absolutely right. Yeah.
Thank you. I tried to play the movie, but I couldn't. Maybe somebody else did.
If you go, let me see. If you go to the slide and then if you put in presentation mode, if you press on it,
Oh,
you should have like a play arrow
okay. I will
on
try.
the bottom,
Okay.
and then
Thank you.
hopefully it will.
Thank you for the presentation. It was wonderful. Really enjoyed it.
Thank
Yeah, I think it was really great. I love this work. Um, just a sort of a general question. This might be unpredictable, but what would you foresee the best possible timeline to make this available to people? Anyway, thanks. I'm done.
Thank you. Yeah, this is we now we are trying to, the regular path. We thought about the definitely what we need to do, and after we complete and we are sure that this de novo design is not revealing another analog that will be better. At the moment, none of them are predicted to be better. We want to synthesize it at a industrial scale, because the formulation for as a pill we already developed, and it's quite straightforward. So after this scale up, we need to complete all the pharmacokinetics and then to fill in for the IND. So as it is a small molecule and not a complex system, our target would be to have at least this in 1 year time to be able to fill in for clinical trials, assuming that we would have all the funding that we need for the scale up. Yeah.
That's exciting. Thank you for the answer and the work.
Thank you.
Hi, Einar. Did you have a question that you wanted to ask?
Yeah, hi, Katerina. Thank you very much. So yeah, this is very interesting to me because it seems that you're trying to prove that what you put into your body actually reflects on the self. Um, what we eat, what we put in our body, and when it comes to liquid and so forth, it's basically information traveling up the spine. It's being thereon reflected by the mind, and thereon actually um, goes through our body as a whole. When it comes to T cells and what actually travels up through the spine when it comes to information here, um, um, the question, and this is for my own curiosity because I haven't studied this field. I do not have the same vocabulary as many of the people in this room, pardon that. But when it comes to the situation when for apoptosis, for example,
Mhm.
there's a driven force of what is being actually, if I can use the word, recycled in our body and what is not. Um, so uh, this is basically, as far as I read your research, goes towards proteins and glucose, not that much lactose, because there's a huge uh, gap here between lactose as a sugar uh, working um, uh, eh, towards other sugar related molecules. But the proteins are important. But what is the driven part when it comes to mitochondria, for example? It's not necessarily the proteins when it comes to how the system works at itself. It's a bit hard to explain. Yeah, thank you.
Yeah, I think yeah, you're right. It's there is a lot that we don't know, and you're right when we say and we are looking into the cell as a cell, and to see the function of these T cells and how they impact the size of the tumor. So we are looking into it at a very high level. But when you touch the sugars, and this is really interesting because For example, in all our work that we are developing with the vaccine in parallel with this small molecule, that our idea was actually to have this small molecule inside the vaccine, where you will not have a true real vaccine, but a kind of a medicine already. It was that what was surprising for us, it was that if we had manos present in this system, we change completely the way that the antigens would be presented, and then the immune response would be activated. So I know that this doesn't answer your question, but in fact, we don't know the answer about the effect on the mitochondria or other, or even in the metabolism of the cells. But we do see a difference each time that we play with some of these metabolites, especially with sugars, that we can drive the immune response in a more efficient way. But we are really far away to understand the full mechanism and also of these small molecules. If you see, we say that they block or they affect the interaction between PDL1 and PD1, but if you see, they bind there, but we also see a decrease on the PD1, and it was supposed to have this effect on the PDL1, but when we do the validation or the control on PD1, we also see the binding, and they are very different. So it's a kind of interaction effect, but it may have as well an effect on the function of the cells that we don't know the origin and how it works, but it works.
Thank you. Thank you.
Yeah, thank you so much for the question and answering these interesting questions. And um, I have a, since I'm coming from the field of neuroscience, I have been also really interested in the immune related connection with some, especially neurodegenerative diseases and so on. And since you're working with small molecules, would you know, or did you test if they would pass the blood-brain barrier? And if yes, would you have some data, or find it interesting to maybe one day check if those compounds could also address different neurodegenerative or mental health disorders? And why I ask is because there's more and more data that shows that mechanisms that change synaptic plasticity, and that neurons used overlap with what is being observed in cancer cells. And there is more labs that are working on using drugs from cancer, epigenetic mechanism targeting drugs to check if in mouse models if that would also treat some mental health related disorder. So anyways, long story short, do you think it would pass the blood brain barrier? And if there would be interest in researching if you could also treat those type of disorders?
Yes, thank you, Katerina. The idea when we were doing the screening, it was to have small molecules that would be lipophilic enough to cross the biological membranes. We didn't do yet a pharmacokinetic study to quantify exactly the amount of the small molecule that we could find in the brain, for example. But this is something that we are currently working, because we are also working in brain metastases of melanoma and breast cancer, and we are now trying to understand if the small molecule alone, or if we need a boost on the immune response to again to increase the amount of effector T cells that we manage to infiltrate within the brain, in order to take advantage of the small molecule. So I from the criteria that we set up in the experiment, I would say that is expectable that they would cross, the biological membranes and most probably the BBB, which is our major target, but I don't have data yet to answer to you to that question. But definitely it is important. We are also working with a group with about multiple sclerosis, trying to characterize the effect on the on microglia and on the myelin. So definitely there is room for. I agree with you. Cancer and the neurodegenerative disease, they will merge more and more.
Go ahead, Joyce.
I was wondering, do you think it has any antimicrobial potential? And also, another question is, would it work in someone who is immunosuppressed?
Yeah, good question. About the antimicrobial potential, I don't know. We didn't test. In the immune suppressed ones, we had some of these tissue samples that were from tumors that are highly immune suppressive. So we see that in those cases, we need to have a boost of the immune response in order to have, let's say, a number of T cells that will be able to have an outcome in terms of the cytotoxic immunity. So these were very challenging tissue samples that we got, for example, from metastases in the bone and also from pancreatic cancer, that they were really a desert in terms of T cells. Even though we saw infiltration of T cells, but the response was much better when we joined, for example, with the activation with CPG, for example, an adjuvant, just to improve the chemo attraction on the T cells.
Okay, thank you for addressing those.
Thank you.
So it's a good time because you just mentioned about the pancreatic cancer. So how was that? Did you use the, for example, gem beside that in combination, or how you did that? That was, for example, some of the research around the xenobiotic transplant, and they are testing those things.
Yeah, this was we have a collaboration with the hospital also nearby our faculty. And these were tissues that were just resected from patients. The patient were not treated yet, so we could characterize the tissues. And the idea was to address, for example, the combination of the small molecule with radiotherapy. So the tissues were irradiated, and then we added the small molecule. And as we were expecting, the infiltration of the T cells and the immune response activation was much higher. But these are really assays that we are just doing, and we don't have yet a validation, for example, in a in vivo model. But we do see synergism. About the drugs, we didn't do yet.
Yeah, I was just wondering maybe you use the gem cytogen or
Yeah,
such a things on the
no,
side.
no, not yet.
Yeah, thank you so much. Dennis, you joined the stage. How are you? Do you have a question?
Hello, everyone. Yeah, I had a question. It was perhaps a little off topic, but we were discussing the blood-brain barrier. I was curious, understanding that the permeability can change, what is the standard size of molecule that can permeate that particular membrane?
Well, there are several factors that I think that will have to be taken into account. For example, not only the size, the monoclonal antibodies, mostly they don't, if you have the BBB not damaged, right? They don't permeate. For example, you can also use these small molecules of this size, like up to 500 daltons, they are expect to, but then it depends on the physical chemical properties of them, like the coefficient, the coefficient partition that they would have. Um, One of the strategies that people use is to use high concentrations of surfactants, so then you can kind of mix your molecules with a BBB, but then you have also some concerns about the integrity of the BBB. But then it depends on the tumors. For example, glioblastoma, you have the vasculature that is not so intact, so it will be easier to get these small molecules into the brain. rather than others that are either non-vascularized or you have the BBB is not compromised at all.
Fascinating. I would not have thought that there would be surfactants used in this. That's really interesting. Thank you so
You're
much.
welcome.
Yeah, thank you so much, everyone, for asking questions, and especially, of course, thank you to you, Helena, for answering all of our questions and sharing your research. And unless somebody has like an urgent question, oh, our star, you have your hand raised. Do you have a last? I'm trying to bring you up. Oh, it worked. Hey, do you have a last question you want to, or a comment you want to share?
Yeah, I do. Thank you. It's concerning breast cancer. I think you said that you used ER positive when you ran your tests, and so obviously there are different subtypes. I'm familiar with the triple positive, so that would have the HER2 positive, the PR positive, and the ER positive. And so I'm wondering if the therapy you're talking about, is this happening upstream of those receptors so that it would work universally for all the subtypes of breast cancer? Or do you have to, are you looking to test further those particular subtypes? You know, because for instance, with the triple positive, there's a thing called crosstalk between, say, ER and HER2, where if you're treating one of the receptors, the tumor can in fact shift to the other receptor to evade the therapy. So I'm just wondering, maybe you said it early on and I missed it, but is the therapy you're dealing with upstream of these receptors so that it would work across all subtypes, or do you have to test the further, you know, do you have to test these other
Yeah,
subtypes?
thank you. No, I think we need to test because actually we had some that, for example, also we were looking first into triple negative breast cancer. This was our first um, option at the time because then you have a higher expression of the PDL1, but also it was reported in some that were R2. So that's why we then tested in both and they work, but definitely it changes the activation of the T cells in the different subtypes. So we really need to check the PDL1 expression and then to adapt according to that expression in these tumors. I don't think it will work universally in these all subtypes that are HER2 positive.
Okay, thank you. But that is a further interest of yours, attempting
Yes,
to
sure.
look into these different
Yeah,
subtypes.
we
Yeah.
are looking into the different subtypes, triple negative, HER2 positive, the different subtypes to try to find a way if we can actually look into the different chemical compositions of these small molecules that may be more related to one than the other, because we are seeing some trend.
Hmm, nice. Okay, thank you for the
Thank you.
presentation. It's wonderful.
Thank you.
Okay, great. Thank you so much for that question and for answering. So yeah, thank you so much, Elena,
Thank you.
for sharing your amazing research with us, and we wish you all the funding and all the smart people that you need to work with you on this, because I think it will save a lot of people's lives, and maybe even, you know, in other disorders, like neurodegenerative disorders and so on. So again, thank you so much for being persistent and always looking from the bright side to improve when you don't get funding. And I hope you'll get a lot of funding. And then we're looking forward to hear more. Maybe you come back and then share more
Sure.
projects and research.
Thank you
That
for the invitation and thank you all for the questions and for this discussion. Thank you. And sure, I'll be really happy to go back.
Wonderful. So yeah, thanks everyone for coming. And if you like discussions like this, follow the club. We have more researchers coming up to share their work with us. Tomorrow we'll have Dr. Lazarov, and she will talk about how she managed to restore memories in mice with Alzheimer's disease. And then on Wednesday we have researcher Dr. Salas, who will talk about model predictive organism design with biophysics and machine learning. And we will have Dr. Varun Venkataramani talking about glioblastoma brain invasion and how that works. That's basically this cancer type invades the brain. And then on Friday we'll have Dr. Goodyear about talking about how he visualizes heartbeats in vivo with the antibody dye. And yeah, it's really interesting. Like it was very cool videos about the activity in the heart. So thank you everyone. Thank you Elena again for being here, and hope we'll talk to everyone soon again. Thank you.
Thank you all. Bye.
Thanks again, Helena and Katarina and everyone.
Thank you.
Okay,
Thank you.
thank you, and thank you everyone, and I'll close the room in 3, 2, 1. Bye everyone.